Welcome to Food Innovation Talk, presented to you by Danish Food Cluster. The topic for today's talk is Bioprotection 2.0 and is presented by Dieter Elsa Graversen, Managing Director at ISI Food Protection, Maja Roe Pelik, Project Manager at ISI Food Protection, and Marta Volpe, Research Scientist at ISI Food Protection. ISI Food Protection is a member of Danish Food Cluster and is located in Agro Food Park in Aarhus, Denmark. ISI Food Protection works close with the food industry in Europe and other regions to help them ensure that their products are safe and stable. We hope you enjoy today's talk, and please remember that questions can be asked in the form. Please enjoy.
So good morning and welcome you to the first Danish Food Cluster Innovation Talk, which is on Bioprotection 2.0. And that's actually some new trends, some new technologies which we want to apply in food to make food safer and more stable with the help of nature. So why do we need such kind of new technologies? And that's mostly because we have, by all these new food trends, also some new challenges. And that could be like what we have now as one of the main trends already a few years, clean labels. So food consumers, us, we don't want to have any E numbers in food. We don't want to have any chemicals in food. So we are looking for alternatives to preserve food. Then we have also health trends like reduced salt, reducing sodium, reducing sugar, having sugar-free food, reduced fat. We also would like to have more and more a milder taste, meaning that we want to have less acid in food. And the food should be also shelf stable. And that's also what we can see more and more here in Europe, that a lot of customers for us asking, can we produce some food which doesn't have to be chilled anymore, which can be stored at ambient temperatures. Another topic is open shelf life. So what happens if the consumer is opening food, and you know how it is at home, it's standing in the fridge and kids coming, getting their spoon into it or their fingers into it. So the food is completely undefined, contaminated, but it has to be still safe and stable. Freshness, of course, that's the topic. Minimal processing, so that you want to treat food animals so harsh. And then, of course, convenience food, ready-to-eat food. But the problem is what we have nowadays, by all these trends what we have without taking out all the preservatives or the chemicals or E numbers, how we say in Europe, reducing hurdles like the salt, the sugar, acid. That has the effect that the growth of some unwanted microorganisms, some pathogenic organisms, or some spoilage organisms could be supported in such kind of food product. So meaning that we have higher food safety risks, or that the food can be spoiled before the end of shelf life. What we do in general is that we distinguish between food safety and microbial stability. Food safety is that we have to be sure no risk caused by pathogenic organisms. So that could be like what Salmonella, Campylobacter, all the other pathogenic microorganisms. So we have to be really sure that we have no risk for the consumers, even so that we have sometimes an interruption of the cooling chain or some other problems during the time when the food is produced until it's consumed. The other part is food spoilage or microbial stability. So we want to ensure that no detectable microbial spoilage will occur until the time of consumption. We have also different scenarios from the time when the food is produced and packaged at the producer until it's consumed. So a standard procedure is, or standard scenario is, that food is produced at a processor, it's packaged, and then it's distributed to the supermarket and finally to the consumer. If it's chilled food, then we have, of course, controlled temperatures, low temperatures, but over a relatively long period of time, which could be even 60 days longer if it's fresh food. The other or the next step would be, if it's what we have already mentioned, if it's opened by the consumer, if it could be recontaminated, if we have some secondary contaminations by the consumer, that's the so-called open shelf life. So we have completely undefined contamination, but a relatively short period of time. The other challenge is, and that's which is coming more and more, if we have catering food which is delivered, here we have just such kind of new robotic system, so we have, so to speak, a black box where the food is transported, and we don't know exactly what kind of temperature conditions we have, or what kind of other conditions we have. So it's a relatively short period of time, but it could be under extreme conditions. But as you can see, we have different contamination scenarios, we have different time temperature scenarios where we have to ensure that the food is not spoiled and that the food is safe. A very good example and that's also showing a bit what the principles are of this biopreservation concept. That's the risks what we have with Listeria monocytogenes in ready-to-eat food. I guess some of you have heard that we have just the biggest case now recently in South Africa, with more than 1,000 reported cases and a mortality rate of more than 25%, which is really tremendous. So it's a really extreme example. But it was caused by Listeria monocytogenes in some processed meat. And what I just want to show you is what the interaction is between the, so to speak, background flora and Listeria as a kind of contamination. If we have, for example, a salami product, and this is just showing some of the results from our, so to speak, data in our lab when we do such kind of challenge test with Listeria monocytogenes. If you have a fermented product like a salami product, we have a very competitive microflora in such an environment. So we add a starter culture to it, sometimes even a starter culture together with a protective culture. And what we can see is that we have only at the beginning, if it's a slow fermentation process, a slight increase of Listeria, but in the further processing, further fermentation, the Listeria count will decrease, and so there's not really a real risk. It's looking a bit different if we have a only slightly fermented product. And this is, for example, and it's a product which is more typical in countries like Germany and Belgium somehow, that this is a kind of minced, slightly fermented product. And here we have also a kind of background flora, which is not that strong as like in a salami, and the conditions also not. So we have a slight increase of Listeria over time, end of shelf life, so that's over 21 days at cooling temperatures. And as we can see, we have an increase up to 1,000 CFU per gram, so this product wouldn't be any more safe. But anyway, the other extreme example is if we have a product, and that's also showing how it happens if we have such kind of food contaminations or Listeria issues. We have some food products, raw materials, they are processed. We are more or less eliminate the whole microflora in such a kind of food product, so we don't have any more competition. And if we have a recontamination, a secondary contamination after the heating, how it happens in reality, so meaning meat, raw meat, processed, cooked, after the next steps, or during the next steps, during slicing until packaging, there might occur a kind of contamination which what could happen. There's no competition. The cell count of Listeria will really explode, and we can get easily Listeria counts up to 10 to the 7th at the end of shelf life. But it's exactly what we are avoiding to do by using some competitive microorganisms. So replacing the microorganisms which could spoil the food by some other microorganisms which can compete, but which don't have the negative impact on taste and flavor, so meaning spoiling the food. What we will show now in the next sessions, and that's by my colleagues, by Maya and Marta, we will show the detection tools what we have to monitor the microflora in food and also to see how the biopreservation system works in reality. And the next part will be to show you some examples of these protective solutions. That would be some solutions with bacteriophages and also with protective cultures.
So before I start explaining about how we can monitor the microbial flora of food samples, it is very important to emphasize that all bacteria are not dangerous, and we don't have to protect against all bacteria. In fact, every single day we ingest a lot of bacteria, and we are even recommended to eat a lot of bacteria to maintain a healthy gut flora. So take, for example, a fermented product like a salami. By the end of shelf life, we have approximately 30 billion bacteria per portion. And this is, of course, due to the use of starter culture in production of such a food. If we, on the other hand, take the processed food that has been cooked, like this ham, we of course have a much lower starting point. So by the end of shelf life, we will have only 500 million bacteria per portion. If we then take, for example, fresh salad like this, of course, the initial microbial load will be higher because we have the natural flora that comes from the water or from the soil of the salad. So by the end of shelf life, we will maybe have 2 billion bacteria per portion. How many bacteria we ingest every day also depends on where we live, what kind of diet we have. So if we do have a diet that contains a lot of processed, then we of course have, like in the American average diet, a way lower intake of bacteria per day than we have in a European diet that contains a lot of fermented food. So this was just to illustrate the levels that we encounter every day, and that we have good bacteria, we have bad bacteria, and we have the ugly bacteria that we want to protect against. Furthermore, besides having different levels of bacteria, we have this huge complexity. And this tree is the complexity and diversity of the microorganisms that are found in our food. It's actually based on the different species that we here at ISI Food Protection isolated over the years. What you should focus on are the colors. The blue and the green colors represent bacterial that are either having a positive effect on the food or a neutral effect. The brown or brownish colored species are of spoilage bacteria or potential spoilers, whereas the red and pinkish colors are the ugly bacteria, the pathogens or the opportunistic pathogens that we really want to avoid. If we take overall, we can say that around 75% of the bacterial species we have isolated so far are actually of positive or neutral impact to the food. 20% are spoilers, potential spoilers, and only 5% are actually pathogens or opportunistic pathogens. Furthermore, it's important to state that this tree is growing all the time. This is not the end picture. Every day or every week we encounter new species. We learn new things about new beneficial bacterial species. And furthermore, we also have the yeasts and the molds, which are important to remember in the whole food microbiota. So besides the level and the complexity, it's also important to remember that there is interaction between bacteria or microorganisms in general in our food. Not only can they be embedded differently in our food matrix, they can compete for nutrients, they can have antagonistic effects on each other, for example, by producing bacteriocins. They can have symbiotic effects on each other, and then they can even send each other signals. So what can we do to monitor this complex and extensive bacterial flora? One of our standard procedures is what we call detect. In detect, we have our food sample. We cultivate on a non-selective media, like this, for example, where we get all the different kinds of microorganisms growing. Based on this sample, we divide each isolate based on colony morphology into groups. And from each group, we select a representative number of isolates that we then send for genetic identification by 16S or 26S rRNA sequencing, depending, of course, on whether we have bacteria or yeasts. If we have molds, we do not send it for genetic identification by sequencing. We have specialized lab technicians that, based on the microscopic and macroscopic morphology, can identify the mold species that we have. The main benefit of this detect method is that we have the species from the food right here, which means that we can deposit them in our strain collection, what I showed you before with the big tree, and we can use these strains for later documentation that a protective solution developed is actually working on the food. So just to give a small example, as the plate I showed before, this can be the result of an analysis of a food sample. Then we select isolates of interest, and we subcultivate them to get them purified. We send them for genetic identification, and we end up with names on species level. Another method for monitoring the very complex microbial landscape in food is next-generation sequencing, also called NGS. This is a metagenomic determination of all bacterial and fungal species in a food sample, and this is done in one single run. Furthermore, it's a culture-independent method, which means it allows the detection of all DNA present in the sample. So in this way, we get a very high-level information, very fast, and at a low cost. And furthermore, it can also detect very low levels of bacteria or fungi present in the sample. We've actually encountered 0.01% of a particular species that was found by NGS. So of course, there are benefits and there are drawbacks of each of the two methods explained, detect and NGS. So the perfect way, or the most optimal way, of monitoring the very complex bacterial flora is a combination of detect and NGS. In this way, we get both the viable cell count, we get a level, how many bacteria, how many fungi do we have in our food sample, and then by the help of NGS, we get the complete picture of all the different species that we have. An example of NGS from also an in-house test was this red smear cheese. We received 2 cheeses. One had a very bitter flavor, taste. The other one was this non-bitter reference. We from NGS, and the result is clear. On the non-bitter reference, we have the protective culture constituting most of the bacterial flora, and on the bitter reference, we have Morganella and Vagococcus, which are definitely not wanted, also explaining why this cheese had a bitter off flavor.
So what I would like to present next is then, after these tools what we have to analyze or to determine the microbial landscape in different items of food, the next part will be how can we protect food if there are some issues with food pathogenic bacteria, so meaning with food safety, or if we have some issues with food spoilage. So what we already do for more than 1,000 years is to use microorganisms for food fermentations. We have a lot of different food which is fermented, different dairy products, tea, coffee, alcoholic drinks, different sausages. In the old times, it was more or less a spontaneous fermentation because we didn't know anything about microorganisms, so we just saw something happens. And we have something which was uncontrolled, but the main purpose of fermentation was to preserve some raw materials. The next step, and that has started, that's already 100 years, and it was initiated in the field of dairy products, that the microorganisms have taken from one batch to the other, and it was the development of starter culture. So it was defined, we have used always the same microorganisms, the fermentation step, the fermentation process, was controlled. The next part, and I think it was starting in the 80s, was the use of so-called protective cultures. And that was mostly using protective cultures in fermented food, and it was mostly, and that's how it has started, to use bacteriocin producing lactic acid bacteria against Listeria. Nowadays we have different categories. I will show you later what kind of protective cultures we have. The next part was the using biotechnological processes, meaning using microorganisms, so to speak, as cell factories for producing different metabolites like bacteriocins, niacin or natamycin, which is used against molds and yeasts. Another category is the fermentates, which is just a fermentation broth, spray dried and used in food. And then also where we also use the fermentation process in a fermenter for producing bacteriophages, which will be shown now in the next slide by my colleague. The bioprotection 2.0, how we call it, that would be now the next step, which is using these kind of protective cultures not in fermented food. It should be also for non-fermented food. And here we have these two different categories, what I have already mentioned, what we will now finally present. First, the bacteriophages, and secondly, the protective cultures.
Yes, I will introduce you to the topic of bacteriophages, and in particular describing what they are, how do they work as an effective tool to eliminate food spoilage, and what are the challenges of using these bacteriophages in industrial applications. So to start, bacteriophages means bacteria eaters. It's a word that derives from the Greek term phagein, which means to devour. And phages are highly abundant in nature. They outnumber bacteria by a factor of 10, and they are also naturally present in food. What is important to understand is that phages have a great specificity. They can recognize and bind to specific receptors on the cell, on the surface of the cells of the bacterial cell, and these make them more specific than antimicrobial drugs. Here you can see an E. coli cell, a bacterial cell, under a massive bacteriophages attack. But what is important to understand is that even only one bacteriophages, which bind to the surface of the cell, can propagate, can enter the cell, propagate inside the cell, and make the cell explode from the inside, and leaving and releasing hundreds of new bacteriophages, as you can see here. What we can do in our laboratories here at ISI Food Protection is to artificially contaminate one piece of food product by using the pathogens that we have in our strain collection, and add the bacteriophages to it and observe if there is any decrease in the amount of bacteria that we add in the first place. Here you can see an example what we do, and what we have done on a study on raw meat. We add a pool of different strains of Salmonella to the product at two different levels of concentration. We then add the bacteriophages and we wait 1 day. What we could observe is that in the treated, in the control samples, the one where we didn't add the bacteriophages, we were able to find the same amount of the pathogen of the Salmonella that we had in the first place. But when we did add the bacteriophages, which are the results highlighted in green, then we were able to detect a very low level of Salmonella, while when we even start with a very low level of Salmonella, we could not detect, we cannot find the Salmonella anymore. So these are wonderful results that we even obtain by using other kind of food products. And all these tests led to the development of a big research project called Top Safe and founded by the Danish National Research Program, GUD. The purpose of this research project is to make solutions against food pathogens available for the producers of fresh products. And that's because the producer of fresh products cannot rely on other methods for preserving the products, like cooling or heating. Otherwise, the product would not be fresh any longer. So having this tool of using bacteriophages, it's actually an important strategy for them. The project started in 2015, and we are currently ending in the last phase of the project. The project started in the group of Professor Lon Bronsted at Copenhagen University, and what they do is to isolate the bacteriophages, understand how they work, and then they pass them to us, that together with other partners like Danish Crown, Flensted, Dampo, Arla, and the Danish Agriculture and Food Council, we test the phages on the actual food product. So, of course, what we did in the lab, it's wonderful and everything works, but it's always a big challenge to go from the laboratory to the indication. There are several challenges in using the phages in food production, and they can be related to improving their efficacy, clarifying their legal framework, and find what are the best conditions for their commercial availability. First of all, regarding the efficacy, it's important that since bacteriophages are not motile, they get to meet the bacteria. So we need to ensure a good enough distribution of the bacteriophages. But on the other hand, we want to avoid that the application costs become too much expensive. Together with this, what we can work on to improve the efficacy is to prepare intelligent phage cocktails composed of different bacteriophages with known characteristic of host specificity. And in this way, we can eat and kill the majority of the bacterial strains which may be potentially present in the food product. Then, of course, there is still the need to clarify the legal situation around the use of bacteriophages in Europe. While in the US, the situation is slightly more clear, and there are already several products in the market which contain bacteriophages. Finally, since the bacteriophages are relatively new as commercial products, we are trying to find what is the right balance between having an acceptable price and the benefits that we can achieve. And this is why we come back to the first point, that it's a critical step to be able to use phages in food production by having a good enough application method on the real food product.
So that was the part what we're doing in the field to protect food by bacteriophages. The other field is how to use protective cultures. So that means living microorganisms, living bacteria. And that's mostly lactic acid bacteria, but also some others. So their role is more or less in food is, on the one hand, to ensure food safety, meaning that these bacteria are able to eliminate, that would be the optimal case if you apply these microorganisms, that they more or less reduce, eliminate some pathogenic organisms, like Listeria, what we have shown before. And the other field would be that we want to improve the quality of food, meaning that we want to ensure that the food quality is acceptable until the end of shelf life, so no spoilage will occur. This requires, of course, that we apply food-grade microorganisms. So that's the reason why we mostly use lactic acid bacteria. And that's now the main difference. What we have already touched is that the protective effects what we have in the food are without changing the characteristics of the food, like it would happen in the fermentation process. Their mechanisms are not completely understood in most cases, but we know that we have such kind of competitive effects, competitive exclusions. That's by competing for nutrients and other essential substances. A lot of these protective cultures can produce some antimicrobial metabolites, would be just organic acids. It could be also some bacteriocins, some other metabolites. And what is relatively new is that we, so to speak, talk with them, meaning that we use this quorum sensing in a way that we send, so to speak, the wrong message, telling them, please don't grow anymore. That would be called quorum quenching. There are different categories in respect of fabrication, in respect of functionality of protective cultures. What is already used since a few years, that's the protective cultures in fermented food, for example, for food safety purposes. And nowadays it's quite common, like for example, for salami products, to use protective cultures to fight against listeria. That's quite standard, and a lot of starter cultures contain such kind of bacteriocin producing mostly lactic acid bacteria strains. And that can be applied for also for red smear cheese from some other products. The other category would be what we have to ensure that food is not spoiled until end of shelf life. And one example is, and that's we have now quite a few of these products commercialized, that's using microorganisms and bacteria, mostly lactobacillus, also in combination with some other species, for avoiding spoilage caused by yeast or molds in, for example, in fresh dairy products. We have just here one example showing that's yogurt, which was contaminated with molds and fermented with and without these protective cultures. It's a very efficient tool to ensure that we don't have spoilage within the expected shelf life. A new field, what we are now more or less stepping into it, which we call bioprotection 2.0, that's using protective cultures in non-fermented food. It's not completely new because we have already some products on the market which are commercialized as protective cultures, for example, for smoked salmon, because smoked salmon, that's a typical product where we have usually issues with Listeria monocytogenes, mostly if it's cold smoked salmon. Because we don't have any reduction steps in the processing chain. So, and this is just one of a lot of different examples how it works by a challenge study, which we have conducted in our laboratories, which is where we have applied such a kind of protective culture in smoked salmon, on sliced smoked salmon, and you can just see here over average of 21 days in the cooling chain how such kind of protective culture would inhibit the growth of Listeria. So the red line, that's the growth without a protective culture within 21 days, and the green line, that's more or less the inhibition effect by such kind of protective culture. A completely new field, what we are just entering and where we do now quite a few projects, is finding some microorganisms which can extend the shelf life or which can guarantee the shelf life, like for fresh cut salads, which we have shown already before. And that would be more or less, or the principle would be isolating some of these microorganisms which are from this food matrix, which are adapted to this food matrix, but without having these kind of negative organoleptic impacts during the shelf life. The next part would be in smoked, not smoked salmon, it's the ham, where we also try to isolate a lot of strains which are there, but which don't have an impact on shelf life, but a positive impact on the competitive flora, on the spoilage flora. So that was, I would say, relatively short, very roughly, what can be done in this field of bioprotection. By having the right methods to monitor the bacterial landscape or the microorganisms in food, to see how they develop, to see what can be done to get it stabilized, and what we can do with, on the one hand, bacteriophages, but also with protective cultures, so with lactic acid bacteria or some other species. I think we have time to answer some questions. So if you have some questions about this topic, biopreservation, bioprotection, just send questions, and I think we are still here, so we can still answer if you have some questions. So please feel free, and thanks a lot for attending, even so that it was for you in New Zealand, Australia, relatively late, but I think it's for us it was really a great pleasure.
Watching this food innovation talk by Danish Food Cluster. You can always visit our webpage for reviewing this food innovation talk. Hope to see you soon and have a nice day.